Join.
You can't.
First person. Join.
I'm not gonna let me see.
I should not join first. If I don't join, other course reps will have sent it to their group, and we're more than 2,000. All those people will join. By the time I now come back after sending it, by the time I now come back before we join, because I like myself. I'll send it.
Momona, you push mute yourself.
Now, so I want question position like, that she does not join. Other questions would have sent the link. It's not like last set of every man for
Everyone charging us and
Okay, welcome back, everybody, and it's glad to have you back. My network, yes, okay, let's continue without wasting time. The class might extend further than 9 a.m. because of the time lost to internet. Okay, so we stopped here. So let's look at carbohydrates. Carbohydrates are long chains of sugars, like I said earlier, and they are made up of, the general formula is CH2O in bracket, and that is in the ratio 1:1 in terms of the carbon, the hydrogen, and the oxygen proportion. Is the ratio 1:2:1. That is 1 atom or molecule of carbon, 1 hydrogen atom, 2 hydrogen atoms, and 1 oxygen atom for a molecule of glucose or carbohydrates. Monosaccharides, there are different groups. We have the monosaccharides. They are simple sugars that are made up of about 3 to 7 carbon atoms. Please note that we said biomolecules are made up of carbon. Then when you have about 3 to 7 carbon atoms, they are usually monosaccharides. We are not saying 3 to 7 monomers now. We are not saying 3 to 7 monosaccharides or 3 to 7 glucose molecules. We are saying when they have the number of carbon atoms in them about 7, they are monosaccharides, and they are either aldoses or ketoses, depending on the functional groups that are attached to them. Somebody asked ketose had the late sometimes. Please, like I said earlier, see on my slide. Okay, there are 2 groups. Actually, we have the acids and ketoses, depending on the functional group that is attached to them. I'm sure by functional group, you know what we mean from your organic chemistry. So when you have aldehyde attached to any carbon on these monosaccharides, they are known as aldoses. Then when the functional group that you see on them are ketones, then they are ketoses. And you know that carbohydrates always end in OSE, O's. So, and these functional groups confer reducing property agents on them. So monosaccharides are known as reducing sugars, right? Because they have aldose, and because they have aldehyde and ketones or ketones attached to them. So if they have aldehydes attached to them, they are aldoses. And if they have ketones as a functional group that is attached to any of their carbon atoms, they are ketoses. And because of these functional groups that are present in them, they are reducing sugars, because the functional groups confer reducing properties or reducing the ability to serve as reducing agents on them. I hope that is clear. Okay, carbohydrates are building blocks, like I said earlier. Carbon, hydrogen, and oxygen, the ratio 1:1, 1:1. Monosaccharides, examples are your glucose, your galactose, and your fructose, right? All of these, what differentiates them? I mean, the glucose, the galactose, and the fructose. You see, they are isomers. Yes, if I can use that word, they are isomers that have different structural arrangements, right? But they are monosaccharides. Okay, the main functions of the main source of energy, they serve as energy storage in polysaccharides. And the example of polysaccharides are your glycogen, your cellulose, and starch, right? And you have them, glycogen stored in the liver in form of starch, in form of in the liver in animals. Glycogen is stored in the liver in animals. It's stored as starch in plants, right? In animals, you see them in the liver where they store it as glycogen, and they are able to be converted to sugar when there is a shortage of sugar in the body. So they can be converted, the glycogen can be broken down into sugar, right? Then they are also found in structure of organisms. They can be as building blocks in cellulose. They make up the cell walls. So, plants have cellulose cell walls. So these cellulose are also polysaccharides. Also the chitin of fungi and your exoskeletons in your insects, your arthropods, right? They have exoskeletons, and these exoskeletons are made up of chitin. So you have one of the features of those insects, I said they have chitinous exoskeleton, right? So that chitin is a polysaccharide. It's a carbohydrate, and it's a polysaccharide. Okay, I said here that the building blocks of carbohydrates are the carbon, the hydrogen, and oxygen in the ratio 1:1. So if you have, okay, let me throw this question open. If a molecule of glucose is CH2O, for instance, and if you have 5 molecules of glucose, how many carbon atoms will you see in 5 molecules of glucose? How many hydrogen atoms and how many oxygen atoms will you see? Okay, somebody said 10 carbon atoms, correct? 10. What about hydrogen? How many hydrogen atoms will you see in 5 molecules of what was it called? 5 carbon atom. Yes. See different answers. Okay. CH2O, 5 molecules. Glucose. Glucose is 66H12O6. So if you have 5 molecules of glucose, how many carbon will you have? 10. Okay. These your answers are funny, but that is what I can see. I see different answers, and they can serve as options when we set objective questions. Your exam will be multiple choice. So these answers, I will put them. So if you see your answer there, you will pick it. And if your answer is not there, you start thinking. If one glucose is, you say glucose is C6H12O6. And I'll say that how many will you see in 5 molecules of how many carbon, how many hydrogen, how many oxygen? Or the formula, what will it be? It will be, if 1 is 6, 5 will be 6 times 5. Okay. So oligosaccharides are formed by condensation of 2 to 9 monosaccharides. Please, okay, enough of your answers, please. Enough of your answers. So thank you. Oligosaccharides are formed from condensation of 2 to 9 monosaccharides units, and there are different types of them. We have disaccharides, we have trisaccharides, and we have tetrasaccharides. Okay, so we said carbohydrates are either monosaccharides or now oligosaccharides, because Oligosaccharides are the ones that have 2 to 9 carbon atoms. If you say disaccharides, trisaccharides, tetrasaccharides, right, you are correct as well, but a general name under them. Monosaccharide is 1, oligosaccharides is what? About 2 to 9. Then you can now have polysaccharides that are more than 9 monomers. So you have the saccharides, example of the saccharides are lactose, your maltose, and your sucrose. And the assignment number 2 for today. Very cheap questions. What are the monomers of lactose? What are the monomers of maltose? What are the monomers of sucrose? In fact, what are the monomers of raffinose? Raffinose is a trisaccharide, right? So what are the repeating units of this raffinose? If raffinose is a trisaccharide, what are the building blocks of what is it called? Of raffinose? Monomers. What are the raffinose? Saturated lipids have only single bonds, whereas unsaturated have a double. You can't hear me. Don't say we, say hi. So we know that you are the only one that cannot hear. Some others can hear me. So say I can't hear you, it might be your network. As long as I'm still here, I'm sure you can hear because I've not moved my mic. If you can't hear me, it's your network. Please don't disturb us with that. Thank you. I can't hear you. I can't hear you. If you can hear me, say hi. We can hear you. Okay, thank you. If you can hear, say hi. Hi, okay, somebody can hear me. Okay, so please, if you can't hear, it means maybe your network is bad. Thank you, thank you. Let the eyes stop coming. Let them stop coming. No. Okay, so I said saturated fatty acids have only single bond in their carbon chain, whereas the double from your secondary school knowledge of chemistry, you see, organic molecules are either what? Alkanes, alkenes, and alkynes. So from that knowledge, alkanes have double bond, right? Alkynes have triple bond, whereas alkanes have a single bond. So here also, you can apply that knowledge, but not entirely. Saturated fatty acids have single bond. So you can picture your carbon, your organic molecules with the single bond in them. So these ones are saturated fatty acids. The ones that are unsaturated have at least 1 or 2 double or triple bonds in them. Then saturated fatty acids are solid at room temperature. Unsaturated are liquid at room temperature. Saturated are fat-based. Unsaturated are plant-based fats. Saturated fatty acids clogs arteries. They are bad. They form what known as bad cholesterol. They are not good for consumption. You are advised not to consume them much or even at all because they can clog your arteries and cause heart-related problems, atherosclerosis, and what have you. Whereas unsaturated fatty acids have the lower blood cholesterol level, so they are good cholesterol. They are good for consumptions, and we have some examples of all these fatty acids or lipids. Okay, so this slide shows elaborates more on lipids. For instance, they are mostly made from carbon. They are repeating units: carbon, oxygen, and hydrogen. Like we have some of oxygen components. Like I said, 3 molecules of fatty acids and glycerol. Phospholipids, they are major form of phospholipid. That is when they have phosphates and lipids, they come together to form phospholipids, and they form the bilayer, the phospholipids bilayer. And you can see there, they have the phospholipid bilayer in organisms. Your cell membrane, the cell membrane are made up of phospholipid bilayer. And what I mean by phospholipids is that there is a head is made up of head and tail. The head is hydrophilic, that is water loving, so it is exposed, whereas the tail is hydrophobic. The hydrophobic tail is found inward and they are hidden. And so before you have them here, let me illustrate where you have your you see here, these ones are the hydrophobic tail, they are hidden, and on the surface is the hydrophilic head. If you can't hear me, please log out and join again. Hopefully you'll be able to join. So the phospholipid takes this structure. You have the head, right, and the tail. This head we say is hydrophilic. It is water loving, therefore it is exposed to the outside, the cytoplasm, whereas the tail is hydrophilic, so it is hidden. It doesn't like water. So that is the structure of the phospholipid bilayer. Okay, let me clear the drawings. Okay, the functions, they serve as long-term energy storage. They are made up of, they constitute the cell membranes of organisms in terms of phospholipid layer. Then some of them are steroid hormones that serve as chemical signals. Your testosterone and your estrogen are all lipids. Then, like I said earlier, the phospholipids, okay, is more elaborate. You have the hydrophilic head, you have the let me illustrate. Here you have the head, the hydrophilic head, and the hydrophobic tail that is hidden inward. And you can see, we said that they form the major component of cell membrane, and the one fatty acid is replaced by a polar phosphate group, which creates the one hydrophilic and the hydrophobic tail. Then you can see here we said our unsaturated fatty acids have double bond. So this can be, the double bond can be seen here. Double bond can be seen here. Another way to identify an unsaturated fatty acid is that where the double bond forms, you see a form of kink or bending in unsaturated fatty. So when you see a kink or a bend like this now, like a bend in this wise, is what is unsaturated fatty. Because where you have those double bonds, they tilt, they bend somehow. So there is a form of kink. We use the word kink, K-I-N-K, to describe that portion, that region that have the double or triple bonds in a unsaturated fatty acids. Then the next one is proteins. Proteins are large molecules that are made up of carbon, hydrogen, oxygen, nitrogen. Then they are repeating units. Their monomers are amino acids, and you have 20 major amino acids, and they are different. You have your alanine, your valine as different amino acids, and these amino acids come together, joined by peptide bonds to form your protein. Okay, then the structure of a amino acid is as depicted here. There are 3 major functional groups, or 2 major functional groups that are seen in amino acid. Number 1, you have the amino group and the carboxylic acid group. Then you have the alpha carbon. The alpha carbon is the carbon that the side chain attaches to. What do you mean by side chain? These amino acids differ, right? Their structure differ, right? So the R chain in all the amino acids, this is the R. So all proteins have a central